Welcome. My name is Katie Kubina, and I'm going to be your host for the Livestream event tonight. We're excited to show you all sorts of great things. We are steaming back from a week-long research cruise to the Northeast Canyons and Seamounts Marine National Monument, which is a very special marine protected area, the only one of its kind in the U.S. Atlantic Ocean. And we're excited to have this conversation with you. So please, if you have any questions, put them in the chat, and we'll be happy to get to as many of them as we can. Earlier in the week, we talked to you a little bit about some of the little things that we found on this expedition. So we're hoping to bring it together all tonight as we head back and show you not just the little things, but some of the big things that we saw too. So let's get started. First up is our chief scientist, Dr. Paola Battalona. Paola, can you introduce yourself and a little bit about what we've been doing here?
Yeah, hi, I'm the chief scientist, Paola Batalona, and I'm pretty much manage when the activities are happening, making sure that we are covering enough space in the monument and sampling enough for all the different things that we're trying to look for a whole view of the biodiversity in this unique place.
Great. Well, when we're talking about biodiversity, we're really talking about all of the life that lives here. And that's one of the reasons this was selected as a special marine protected area, is because it was considered a biodiversity hotspot. So lots of life living there. So we're all here to figure out what's living here, why, what all that life is doing. How do we get started? So what's one of the tools we need to use when we're getting started?
Well, we could start by acoustically finding out where things are, and then we can target our sampling based on where animals are hanging out or where we see more, diversity based on some different equipment that we're using it on board.
Why don't we go ahead and take a look at some of those maps?
Yeah.
So we're here now inside the dry lab. And if you can take a look here, here's our coastline here. So here's the Northeast. You can see New York, Long Island. Up at the top over here is just a little bit of Cape Cod. And here's our track. So here's where we went. We left from Yukon Every Point campus. We're on the Research Vessel Connecticut. And we steamed about 130 miles out to Oceanographer Canyon here. And then we went on to Bear Seamount, back in, and then we hit another canyon. You're probably wondering, you probably can imagine what all these different colors are. So typically this is a temperature map. So blue is cold, red is hot. So this over here, the snake-like red thing is the current, the Gulf Stream that comes through here. So we were really surprised when we left. There was like a cold spell, and then when we got out to the site, it was like 80 degrees, the water. It was really wild. We weren't really expecting that. At least I wasn't really expecting that. So there's our temperature map. There's our location. Here's a really great map. This is a bathymetric map that shows our locations. And the way to think about that is if you took the ocean and you drained all the water out of it, then this is the sort of features the geologic features that you would see. So this is the first canyon, Oceanographer Canyon, which is actually like twice as deep as the Grand Canyon, which is pretty interesting. Then we went to Bear Seamount here. This seamount is an extinct volcano. So as the continental plates shift over a hotspot, it makes these volcanoes. This is the oldest. Bear is the oldest seamount, the oldest extinct volcano. They think it's about 100,000 years old, and its depth is actually it goes down, so it's below the surface of the water. But if you were to look at the height of it, it would be actually about 2,000 feet higher than Mount Washington. So this is a very large underwater mountain. So these geographic features and geologic features is what gives rise to all of the interesting like currents and upwelling. And that's when you have cold water bringing nutrients up to the surface. And that's what all the animals love, right? They love all that nutrients, which feeds the base of the food web. Right. So I know you study a lot of that, but you have a very special technique, and this tool helps you a little bit with that. So maybe you can start with this, and we can go outside and look at the tools you use.
Yeah, for sure. So here we have a split beat sonar that uses sound, like almost like a bat or a dolphin will use some pings to find their prey. And here you can see that this is a transect across oceanographers. This is when we started. And you can see that the sound starts traveling to the bottom. This is the bottom of the canyon and then starts dropping off. And not only can we see the bottom, but we can see a couple of features like this light blue color. That means that there's some animals hanging out like around 200 meters when we look at the scale. And then other portions of the water column show 50 meters where other animals are hanging out. So now I know where to sample. We know where animals are hanging out, so we want to find out who they are.
So let's go outside and you can show us your nifty tools for doing that.
Yes. So as Katie mentioned, we're in transit and we're not on operations. That's why we're not wearing our hard hats, but it's always important to have our PFDs when we're outside. This is a CTD or a rosette. This rosette has some sensors that tells us the salinity and how deep we're going. And when we send these bottles, now that we know where the animals are hanging out, we can tell from the computer where to close these bottles like this. So let's say I say 200 meters, and then eventually it will close. And then I have water from 200 meters here. And since there were animals hanging out, they might have left a lot of their DNA. So then I study the environmental DNA. So I take this water out with a lot of gloves. Right now, we're not doing the real thing, but usually we have to be very careful not to contaminate with our own DNA. And then I'll bring my water samples to the lab. Would you like to come?
Go ahead.
All right, let's go. So this is the bubble. Welcome to the bubble. This is to keep, again, the DNA from the humans outside from our environmental DNA. So we bring the bottles that containers like this have all the water from different depths that we think are interesting or have animals. And then we put some tubing, and this is our pumping system that will filter into small filters. It will concentrate all the DNA. And then those filters, we put them in liquid nitrogen because we want to make sure that the DNA stays stable and it doesn't break down, it stays in one piece. So here is where we keep our DNA once we already concentrated in all those filters. This is liquid nitrogen, which is like 10 times cooler than your regular freezer. So we are very careful when we use it. This is how we keep our DNA safe. And that's, yeah, once we have our DNA, we go to the lab, we extract the DNA, and then eventually the goal is to have all the sequences from that environmental sample. And each of these sequences, just like the barcodes of your groceries beep at a scanner, we can use those sequences of barcodes for each of the species that were hanging out around the features that we thought it was interesting, like 200 meters. And so these barcodes that you see on the screen are gonna tell us what kind of shark we saw, maybe a jellyfish, what kind of fish. So I'm looking forward to seeing all that data and all the sequences when we're back at that.
So Palad, will this help you determine species big and small, like the whole range?
Yeah, we'll be able to detect, with the markers that we're using, we'll be able to detect from phytoplankton all the way to seabirds.
So you can see the entire food web in just a little bit of water. That's really incredible.
Yes, super excited to find the results.
All right, well, there's some other scientists in here I'd like to introduce you to. Here we have Dr. Mary Beth Decker and Kylie Chernesky, and they have been studying gelatinous zooplankton. So maybe you can tell the groups a little bit about what that is and how you collect it and study it and why.
Sure. Kylie and I are studying gelatinous zooplankton, and there's a couple ways that we're doing this on the ship. One way is with something that we call a gel net, which is a very large opening net that it has mesh that allows a lot of the small things to go through, but then we can capture the gelatinous species, and then that will filter down into the cod end at the end of the net, and then we bring this cod end into the lab where we can gently pour it into a bucket, and then very carefully, because these are very fragile organisms, we spoon out individuals, and then we may look at them under the microscope in a petri dish. We may take a little sample of DNA so we can analyze their makeup, their species makeup, and we can then get a better idea of what the gelatinous predators and prey are out here. So Gelatinous zooplankton are interesting because they are food for many fish, also sea turtles, and even some seabirds. And they're also prey for many of the fish and other animals that we have here in the food web. And Kylie will tell us about a second way that we study gelatinous zooplankton.
Yes, we also have been studying the gelatinous zooplankton through an underwater camera, which is called the plankton scope. So we can put the plankton scope out on the back of the boat, and it takes many pictures per second, 10 pictures per second, and we can then analyze to see what animals are in those pictures. And it's great because it takes pictures of the plankton in their natural environment, so it won't get damaged. Some of the gelatinous plankton are quite fragile, so they can be damaged through a gel net. So here are some of the pictures that we got through the camera. This is a new picture. It's a comb jelly. You can see the what's called cilia, these lines. It kind of looks like a comb, a tooth comb. So that's that plankton. Here's a krill, which is a crustacean. Some copepods. So this is a copepod with some eggs at the back. This is a radiolarian, which is quite small, so the plankton scope can capture very small things. Another krill. Here is a jelly, and this is another jelly. And this last one is a baby jelly, so it doesn't have those tentacles yet.
So it doesn't end here, right? There's a lot more work once you get back to the lab.
Yeah, that's correct. We'll be going through the plankton samples and doing a more thorough job of identifying the species that we have here. And Kylie will be working with the data, extracting images using artificial intelligence and machine learning to capture images from all the many megabytes of data that we collected.
Thank you. So I want to pause here and go see if we have any questions. I know Whit is at Mystic Aquarium looking through all the questions we're getting into the chat. So Whit, are there any questions that we can ask for
Yes,
our group
Katie,
here?
we do have one that a couple of good ones. One is, does a rough weather, if you all see that on a voyage, does that compromise taking any of the kinds of samples? Maybe the eDNA or the gelatinous zooplankton?
Yeah. Well, that is a great and relevant question. It's like, does the weather impact what we can do on the ship? So does bad weather sometimes compromise what we're able to accomplish on a cruise? I think we have some living examples of that on this cruise.
Yeah, for sure. We had to pivot and sometimes do more of that acoustic runs instead of like having gear on the board outside. We can't really do a net that is so heavy. The plankton scope is really heavy and we want to make sure that everybody stays safe. So we definitely pivot, but we still were able to cover a lot of different sampling, which we're very satisfied and excited to share.
Yeah, so on this particular research cruise, we were hoping to get to 4 of the features in the monument, 2 canyons and 2 seamounts. We ended up getting chased into hiding around Nantucket for a day because the seas were just going to be way too rough. And not only is that difficult for putting equipment over the side, it can be dangerous, and a lot of us don't feel really good if it's that rough.
So,
Great question. Any other questions out there?
We did have another one. Do you all have a favorite zooplankton? Gelatinous zooplankton.
What is your favorite gelatinous zooplankton?
can I go first?
Yeah, go ahead.
Salps. Salps are my favorite. Okay, I'll share.
Salps are cool, but I like these siphonophores here.
What is a siphonophore?
Siphonophore is a colonial animal. So it's actually one animal that's made up of many different bodies that are genetically identical to one another, but they work together in concert and they have different jobs. So that large float that you're seeing at the top is something that it uses for flotation and jet propulsion. But these trailing pieces behind it have different jobs. Some act as stomachs and mouths, and others act as reproductive organs.
Very cool.
Very unusual.
Well, we're going to have to move on, but thank you so much. So let's go back out on the deck and we'll see who else we have out there to speak with. There you go.
Thank you.
Hi, so we're here with Fafa. Fafa is a graduate student at Duke and is also interning with NOAA Fisheries this summer to do work related to the monument. So we're really excited to have her. And she got like right into the thick of it, like any good graduate student should do. She wanted to do everything and was totally brave. We worked together, Fafa and I worked together with a team of people to deploy and execute Dr. Peter Auster's research
That we have in these PVC pipes. But yeah, so that's the drifted ones. We also have the URI brub. Oh, and there you can see.
And that was sort of a gift
Yeah.
from a PhD student, Christine De Silva.
Yes.
And so we had no idea, like it was first for all of us to deploy this thing. It was a little scary, right?
Yeah.
But we did it.
And I think what you're seeing right before then was the types of bait we put in all the brub systems, regardless of whether it's the URI stationary one or the drifting one. What you'll see here is the type of mackerel that we use and the literal cutting board that Katie has used to trap all the bait. We'll use a can of sardines and put all of the good smelly fish in here before it enters this container that gets strapped on to the various brubs that we have. Sorry, it's a little rocky.
It is. This is a key component too. Maybe you want to
Yes.
show that.
If you can see, this is the housing system where we have a GoPro camera that is basically set to be in the water at either 20 meters, which is what the drifting brub does, or a little bit farther below for the shark cam brub. But this is what records all of the data and any apex predators we might see.
Do you want to show us the shark cam device?
Yeah, definitely. So the shark cam is also a brub. I think the biggest difference is that it's deployed at night, which means that we need things like this, high resolution flashlights. And if we come across over here, you'll see that the contraption is a bit bigger than the drifting brubs that we're using. So imagine that there is a camera set up right over here. And what we do is put the lights flashing towards the lens. And once they're on and the camera's on, we're ready to go.
So we've got that, and then we attach the bait PVC right there.
Yes.
It gets lowered. It was a little scary.
Yeah.
That one time it gets lowered into the water. We did this once at night and once, I think, during
I think
the
we
day.
did it both at night.
This one is a night
Yes.
device, right?
Both
So
at
there's
night. And
day brubs and night brubs.
exactly. And unlike the ones that drift with a buoy, we actually attach it to the winch and let it tow with the boat at pretty low speeds as well. But it's just a little bit different of a contraption, so we can adjust the depths that we have the shark cam on.
And how do you retrieve them from the ocean?
Great question. The winch will bring back the shark cam kind of to the surface, and basically there'll be two people with hooks attached to a ring, a rope, and you kind of go fishing a little bit. You grab you can see right now, you grab one of the corners of the frame with the hook, and then that attaches some type of rope. And that way you can stabilize it as the crew team brings up the A-frame onto the boat.
Of course, when this is going on, we're wearing our hard hats.
Yes.
Like we're being super safe because they take the chain off for that because you're having to hook it from the ocean. We usually ask the people with really long arms too to help out. So it takes a village to get all this stuff safely done on the ship, but it's lots of fun.
Yeah.
Are there any questions for Fafa before we move on?
Well, we have an interesting couple that kind of go together. How deep is the water where you are now? And can we also ask, how does the depth affect the zooplankton, phytoplankton, and the larger animals that you are seeing? Is there a generalization across depths?
Well, I think that's a good question. I don't actually know exactly what depth we're at right now, but what I will say is that when animals, like the smaller animals like plankton and stuff, tend to go through this like vertical migration during the day and the night. So you can see them at this, like in that map, like a scattering layer, and it helps you figure out where you're going to sample. And where the little animals go, you often find the big animals. So when we were crossing over Oceanographer Canyon, which is right along the continental shelf where a lot of things happen, we saw this like burst of activity. We saw lots of seabirds, we saw whales, we saw dolphins. So you can see where everything is congregating around. They're congregating around the food. So life attracts life, I guess we would say. All right, thank you for that question. We're going to go up to the top deck and visit our seabird survey team. So let's. Oh, thank you. Just saw some seabirds. Hi, so we're here with Patrick Moffett, and Patrick Moffett is the superintendent of the the Northeast Canyons and Seamounts Marine National Monument. He and Troy have been doing amazing work doing seabird studies. Why don't you tell us a little bit about yourself and the monument and just how you ended up in this interesting place that you are now?
That's a good question, Katie. For me, I've been looking back and had time to reflect this week. This is kind of a lifelong conservation journey, and I didn't recognize as a young kid. But I grew up right around the corner from Blackwater National Wildlife Refuge on the Chesapeake Bay, and that was a place that's near and dear to my heart. And eventually, it ended up working in this field. I found myself working in California's Great Central Valley and managed wetlands there for 10 years, and then eventually moved to Fish Springs National Wildlife Refuge in Utah's West Desert. So you might think with that background, how would I end up in a place like this? But during this reflection this week, I've had that time to really understand that there's a lot of connectivity amongst those places. One in particular being that a lot of them are built on, a lot of the work that we do is built on partnerships. And that's really what I've gotten to see up close here this week. Again, this is my second time out to the monument, my second year here. And it's really been a remarkable thing to watch this partnership function. There's 9 different representations here, all working together around the clock. We've even had people falling asleep at their table, working overnight. It's been really impressive to see how functional an effort like this can be with the right people in place and the right partners in place. And so that's really what I've taken away from this week and that time to reflect, is that you kind of wonder how you get to these situations in these amazing places, but then you also see the collective group that makes all that happen. And that's really what we're witnessing here today.
I was sort of a little surprised to see that there's more connection than I would have thought too. Like, it's a really remote area, but we did see some species that people are familiar with more inland. Maybe you can talk about that when you're talking about the bird surveys you've been doing.
Absolutely, yeah, and I'll let Troy share some more on that. But that's one of the key things that we really wanted to emphasize, not only with this effort at recognizing the value of the monument itself, but also starting to build that understanding of connection between this place and the coastal places in our region. There's refuges all over our coast: Monomoy National Wildlife Refuge, Ninigret National Wildlife Refuge, Parker River National Wildlife Refuge, Maine coastal islands, all the way down to Blackwater. This place is in a straight line, not too far from all of those places, and the species that are using those areas often overlap with where we are now, and that's not well understood. And that's also part of the scientific effort that we're trying to really understand better now, but also connect to the public and really relay that message that this place, along with being a marine national monument, of which there are only 5, it's also a national wildlife refuge, and there's 570 of those across the country. Many know their local ones in their area, but they don't realize that that's a collective system. And, that's really, to me, the piece that I want to emphasize, is that connectivity between those places, the people. Hopefully what we're showing you, this gives us an opportunity not only to show the science, but to connect back to the public that invests in these places. And this hopefully this shows their value. And that's been a really wonderful thing to be a part of for these last 10 days. Last year when we were out, we had some real challenges with weather and equipment, and we got a lot of good work done, but it was really challenging. This week has been a really well-functioning machine, and it just feels really good now that we're kind of wrapping that up to have a great week of accomplishment. So we're thrilled to now bring that to you. So thank you. And I'm going to pass this over to Troy so we can kind of explain a little bit more of our bird surveys themselves. Okay?
Thank you, Patrick.
Good evening, everybody. I'm Troy Wilson. I'm the inventory monitoring coordinator for the refuge system in the Northeast, and I'm thrilled to be part of this team. I'm going to talk a little bit about what we're doing, why we're doing it, and the birds that we're seeing. I've worked for the service in different capacities, but it's afforded me the opportunity to work with waterbirds and shorebirds for a good portion of my career. And now I get to explore the world of seabirds. And it wasn't until I got this job in the Northeast region, learning more about the resources that we have, like as Patrick mentioned, on the Maine coast, and all of the resources across the Atlantic seaboard. It's been amazing to learn more about that aspect and seabirds specifically. So what we're doing is we're recording observations as we go along with the operations of the research that's happening on the vessel, and we're taking photography and submitting as detailed species observations as we can get to on eBird, which incidentally is the world's largest biodiversity-related citizen science effort. And they just celebrated passing 2 billion bird observations, which is incredible. And so we're submitting to that effort as part of what we're doing here. We have this really fancy camera equipment for you photography nerds. This is a Nikon D850, and we can go from 200 to 500 zoom, and mainly we're doing it for bird identification. We're looking for subtle nuances in plumage or morphology that helps us get down to the species level. And so what we've been seeing mainly is a group of birds called shearwaters and storm petrels. So I'm going to talk about the shearwaters first. Shearwaters are just an amazing group of birds that we're seeing here in the monument, but have traveled across the hemisphere from the waters off of South America and Africa. They travel hundreds of thousands of miles to in their hemispheric journey through their migration, and then we get to see them here. Their behavior is incredible. They glide over the surface of the ocean, inches above the water, kind of tracking swells and waves as they're foraging for the fish and crustaceans that they're after. And it's just incredible to watch and learn about. And thus the name shearwater. So they're just cutting and gliding through over the surface of the ocean. It's incredible. The other group of birds that we're seeing a lot of are called storm petrels, and there's a lot of mythology and lore around these birds. Sailors often thought that they were bringing or signaling changes to the weather, and they do that, and thus their names, storm petrels. They often will signal changes in the weather, but these are tiny. As opposed to the larger shearwaters that we're seeing, these are tiny sparrow-sized birds that are just fluttering. Their flight patterns look much like butterflies, really, as they flutter across the surface of the ocean. They're often looking like they're dancing on the surface of the ocean, and it's just a wonderful group of birds. And to speak to the connectivity that Patrick was mentioning, the storm petrels will travel hundreds of miles offshore, feed all through the night and into the day, and then travel back daily to feed their chicks that are nesting on the coast of Maine in their burrows, which is just incredible to fathom. So I'm just thrilled to be part of this effort, learning so much. I'm so inspired by everyone here and what I'm seeing. And to speak to that, I just want to talk a little bit about some of the oddities we've seen. My background is largely in land birds, but at one point we saw a purple martin flying over, and that was unexpected. Later in the season, and this often happens on ships out at sea, you'll see small land birds stop and rest because many of them are migrating over open ocean, which is in and of itself also an incredible phenomenon. But anyway, we saw the purple martin. We've seen some other things that were unexpected, but this morning I had a great shearwater land almost at my feet, which was really exciting and unexpected, and almost as if to say, as we're departing, you know, thanks for the visit, safe journeys home. So thanks for the opportunity. We're thrilled to be able to bring this amazing place to you and I'm just, it's a privilege and honor to be doing this work. And so thank you.
Thank you, Troy. And thank you, Patrick. They're selling themselves a little short too, because you can hold on to that, because they were also the ones who were like first line of defense for spotting some other interesting things like the marine mammals in the monument. And, you know, there are other entities that do aerial surveys like New England Aquarium to survey the marine mammals. And we really got to see up close just how rich this area is. And it really, you know, goes all the way back to those like tiny little planktons all the way up. It's a lot of the whales feed on plankton, but it is also so important as the base of the food chain. So you see this really rich ecosystem. Are there any questions for Patrick or Troy? Or Patrick and Troy?
Yes, we did have a couple of great ones. The first is Troy mentioned the eBird database. Can regular folks contribute sightings to that as well?
Yeah, that's a great question. You mentioned the eBird database. Can regular people contribute to that as well?
Absolutely. That's the key. Absolutely. That's the key in citizen science. And that's how we've been able to track and learn so much about migratory patterns and population changes. So yes, you can sign up at eBird and you can have your own account and it tracks your life list. It's revolutionized birding in many ways. So yes, absolutely. Check it out. It's housed out of Cornell University, Laboratory of Ornithology.
Yeah, and beyond eBird, there's a lot of ways that people can join community stewardship projects, help restore wetlands. So you can help these places out here by helping closer to home and work on eco restoration projects. Any other questions, Whit?
We did have another great one. The seabird numbers above the surface, do they tell you anything that's happening below? Do they kind of predict some of the other tests that you all are running?
Yeah, that's a great question. So when you're seeing seabirds on the surface, is it an indicator of what's going on below the surface?
It's interesting that you say that because this was an opportunity to this week, and we, you know, with all the other scientific efforts going on at the same time, we were often able to kind of run from the deck when we would see larger groups of birds and then go down and check and see what was going on below. And that was a really interesting bit of conversation. I mean, you know, we're still at kind of the foundational level of this, of learning those interactions. But certainly this week, there were several examples where we were seeing birds on the surface, and that would tie to the marine mammal activity we were seeing below. And you know, it's interesting to see that. So, I mean, that's the value of the trips like this. You can accomplish some of this through other scientific efforts, but being out on the water and actually seeing it in action is so helpful, especially when you can tie it to the rest of the science that's occurring on the same vessel. It's really great to have this particular opportunities like this.
Yeah, there's really no substitute for being here in person, right. Seeing it all, right. Getting a chance to interact with your colleagues, testing hypothesis. It's an amazing opportunity. We're going to go downstairs back to the deck and take a couple more questions. So let me take that from you. So Whit, if you want to give me a question that might be suitable for any number of members of our team, that would be great.
Well, we wanted to ask, what have some of the folks' favorite moments been on the trip so far? Really, for anyone.
That's a great question. So what has been some of the favorite moments on the trip so far? Let's start with you, Kylie. We'll start with our young people, our PhD students.
My favorite moments have been when somebody spots a marine mammal and then they'll put in the group chat and you have to quick run to the top at the bow and you can see them. And it was really magical. I've never seen whales in the wild, and maybe not even dolphins either. So that was my favorite.
Yeah. Why don't you pass it to Bafa?
I might have been thinking about saying the same thing, but I really loved getting to know all the different project names going around such a, not a small vessel, but like in such a tight space. It's been really incredible to see the team run and check in with each other, and have ops running night and day. And my second shout out is the plankton or the gel nets in the evenings over Ladonia Canyon have garnered a lot of salps, and that has been really fun for those who are awake at those hours, checking out the specimens that Kylie and Mary Beth are collecting.
Anyone else want to answer that question? It's a good one.
I could add one more piece. I mean, I think going back to the question we had before about, you know, the connectivity between the birds we've been seeing in below. I mean, we had several shots actually going through our photos, and I can't wait to look at more where we actually could see marine mammals and birds in the same photos. I mean, that was really cool to see some of that visual interaction actually capture in real time.
How about you, Paolo?
Yeah, I think seeing everybody excited about the different parts, and even people that are not familiar with, you know, the eDNA, when I see the filters that are full of green and we're seeing phytoplankton and then a lot of zooplankton and a lot of seabirds, it's like it's coming all together. This is why we're out here, to find out more about this diversity.
Thank you. Anyone else? Troy?
Aside from a bird landing at my feet, I'm going to just say that we got 2 observations of a great skua, which is a life bird for me, and that's always thrilling. And it's a big gull-type bird that was unmistakably like that's what that had to be. And we got good photos, and we're able, and then we geeked out on the identification. So that was a pretty cool moment for me.
How about you, Mary Beth?
I really enjoyed this cruise. I've enjoyed meeting new people and working with people. We're going home now, and I'm starting to feel like I'm going to miss my ship family.
Yeah. Well, I think I'll round that out since we're running out of time. One of my favorite moments, other than all of the things that have been mentioned, is the beautiful sunrises and sunsets that we get here out on the ocean. You really get a sense of what a magical place it is. We're getting a little bit of a sunset right now. So before we close it out, I want to give a few shout outs. One is to the amazing crew on the ship. There are 4 crew members and a cook, and they're all fantastic. They work 6 hours on, 6 hours off. This ship runs 24/7, and they've been amazing. Also, Mystic Aquarium and Yukon as the co-leads of this. I want to thank Yukon for giving us access to this amazing ship. The RV Connecticut is owned and operated by Yukon Marine Sciences. There's all of our partners. There's Duke University, University of Maryland, Yale, of course, U.S. Fish and Wildlife, and NOAA Fisheries, who co-manage the monument, and the URI's Inner Space Center. I'd love for us to get a quick look at our videographer, who's done outstanding work. Can you turn the camera on yourself for a second, Alex? Big shout out and thanks to Alex for doing such great work on this ship and helping to bring all of this special place to all of you. So thank you for tuning in, and please continue to follow along on the expedition landing page at mysticaquarium.org. We are going to be posting another social piece and also a wrap-up reel where you'll get to see all the stuff in action. So thanks very much. Goodbye.